Okay, hi everybody. We're gonna do kind of a review video for our latest lecture, the mitosis and the cell cycle lecture. We're gonna just start with mitosis since, oh shoot, give me this thing. There we go. We're gonna start with mitosis since that's the table you want to fill out still, and I kind of wanted to make sure we went over that maybe a little slower, that you can pause, things like that. So this initial image here is a good overview of everything. Essentially, the goal is to rip this DNA in half, go to each cell. A key thing that we'll learn, especially when we take a look at the meiosis, is that each of these things being taken upwards towards the new cell, these are copies of each other right here. And so, for example, this is going to be like maternal chromosome 18. And like this could be paternal chromosome 18. So the maternal and the paternal in mitosis, they're lined up like this could be maternal, this could be paternal right here, and that was maternal. So it's always going to be a straight identical copy in that split. So that's something that we'll see, especially in in meiosis, that we're not going to be doing. So for mitosis, yep, identical DNA splits. So the way that we kind of I'm trying to teach you on this is that the microtubules are kind of the key, and these are what are forming that spindle. They are what going to be attaching to the chromosomes, to the cell itself, and pulling and pushing in each case. And they're just made of these two small little protein units, alpha tubulin and beta tubulin. And basically every cell has tons of these, and that's how a lot of these cells move and go about their business, not only in mitosis, but this is a key component of the cytoskeleton. So if you take cell bio in the future, you'll see this again. Biggest thing with these microtubules is, remember, they're just made of these little alpha and betas. They can go down and they can shrink. They can also grow and go up, for example. So that's how we have the like back and then forth type of stuff. Well, that's weird. Okay. So these things can go up, they can go down. They're really fluid. So we're looking at these three keys, and these are in your table, and they're a good way to understand what's pulling and what everything is doing. So everything is starting from this microtubule centrosome organizing center. On your table, this is the centrosome. All the microtubules are going to be coming out of the dual centrosomes that are on either pole of the cell. So think of like a north and a south pole, and like this is your equator, for example. Because the whole goal is to rip this half, this copy of the chromosome, to rip that this way, for example. And remember that these are two identical copies, copy 1 and copy 2. And so you want to rip them this way. You want to rip it that way and that way. And these microtubules, they're going to be the ones doing all of that. So let's start here. All right, so at your interphase, what's happened is that the key thing is that your chromatin or your DNA, because remember chromatin is just like tightly packed DNA, this has been duplicated. So now you have two copies of a maternal chromosome and two copies of your paternal chromosome. And remember, the maternal and paternal chromosomes are homologous, as we can call them, so they're pretty much the same. There's just a few variants between the chromosome your mom gave you and the chromosome your dad gave you. But at this point, you essentially have 4, because you have 2 copies of the mom, 2 copies of the dad one. So at this stage, this is right when you're going to duplicate your centrosomes. And these are the 2 ends of the spindle, and you see them like start coming around here. Your nuclear envelope is intact, plasma membrane's good, everything's fine. So prophase, and I made this still count as prophase, as prometaphase. What happens is that the nuclear membrane is starting to disappear. It's starting to go away. All your DNA is starting to condense. It's condensing quite a bit, and it's going to condense into this chromatid structure that we see that can be split 2 and 2. And we saw that these 2 are all, they have duplicated at this point. Okay, so we're still in prophase. So what's happening right now is that the centrosomes have found their poles. So that's something you can write in your table. Centrosomes have found their poles at this point. What is happening with the astrals, so AMT, astrals are grabbing the back of the cell. Kinetochore, microtubules have now found and grabbed some of the DNA at the kinetochore. So remember, that's just this little structure right here that they're grabbing. And then some of the polar microtubules, you can see two happening right here. Some of the polar microtubules, those are the ones that are attaching from the other side of the spindle. They're not attached to DNA or the cell, they're attached to each other. Okay, so at this metaphase point, we've basically kind of gotten ready and attached all those microtubules that we wanted to. And the DNA is like fully condensed, fully copied, everything's good. Okay, so metaphase is easy because all this is that everything is perfectly aligned on this equator, essentially. Everybody's perfectly lined up, and nothing's going to happen until all 23 copies of chromosomes, and in this case, actually, you're going to have 46 of these little things right here, because remember, there's 2 copies of all the 46. One's going to go that way, one's going to go down. They're all lined up, and nothing's going to happen until all of these KMT, so the kinetochore microtubules, are attached to DNA. Because that's a key checkpoint, is that all these have to be attached first. So at this point, DNA is fully condensed, nuclear membrane is completely gone at this point too, so don't worry about that. Astrals are still on the side of this, on the top of the cell. Polars, you can see right here, are attaching to themselves, and the kinetochores are attaching to the DNA. At anaphase, this is when we rip everything apart. Here's one identical copy of a chromatid. Here's another identical copy of a chromatid. They start going different directions, and they're going to start splitting, and that's anaphase. That's the pull. Okay, so at this point, astral microtubules are pulling down. They're pulling towards the cell. They're pulling that direction. Your kinetochore microtubules, depending on the direction, let's start here, they are pulling down as well. I cannot get that. They are pulling down, so kinetochore microtubules are also pulling this direction. However, your polar microtubules, the ones that are touching each other, they are pushing each other. They're pushing each other the opposite way. I don't know how to control whether it does that. Okay. So at this stage, see this little connection right here, they're going to be pushing the opposite way. And so these two are actually going to be pushing each other kind of like in this, like, kind of in like a cross, like push like this. So as long as they push each other both directions, the force is going to be exerted to split the cells, essentially. So at this stage, at telophase and cytokinesis, the actual membrane starts splitting, the nucleus starts coming back, and nucleolus is like a little bit of the nucleus. We haven't covered it, but you can cover it later if you want. But yeah, this nuclear envelope is going to start forming. The DNA starts decondensing again, and that's important too. All your microtubules at telophase and cytokinesis have also gone away, you can tell. The little centrosome is just going to hang out here and wait for this new full cell to divide one more time before it duplicates itself and does it again. So those are the keys of mitosis. Hopefully this video is comprehensive enough for that table. Definitely let me know if it's not, and we'll go over a little more. So this is just an interesting slide. This is how microtubules are moved, is that there are motor proteins that essentially they attach at the kinetochore. So this purple right here is DNA, and the kinetochore itself is actually a big motor protein. And when it's time to pull towards that bottom of the cell, the motor protein is going to start pulling itself along this big microtubule back towards the new chromosome, and it chews off those little bits of alpha and beta tubulin as it goes. So that's just supplemental if you want to take a look at that. Okay. All right, so now let's go back up to the beginning. Let's click this. And we can cover just the cell cycle in general. So we had some good time in class to cover this, essentially, and we can just cover the checkpoints right now. Essentially, at G1, so we've just finished dividing, so remember mitosis is going to be the final split right here. So say that we're at G1, we're kind of going to decide if we want to divide again. Do we have the nutrients? Do we have the cellular contents? Do we have all the chromosomes? Is everything like at the basic level pretty good? If everything at the basic level is pretty good, we can move on and we can decide that we're going to synthesize and copy each of the 46 chromosomes. So the 23 from mom and the 23 from dad now each get their own copy. So they're identical copies, the mom pair and the dad pair. Because usually you just have, say, chromosome 7. You have one chromosome 7 from mom, one chromosome 7 from dad. But at this stage, you have 2 from mom and 2 from dad that are exact copies, because the one from mom and dad are not exactly alike, but the chromatids that you copy here, they are exactly alike. And the reason for that is that during mitosis, we want to have exactly alike cells that split off here. Okay, lastly at G2, this is after you've done all your DNA and DNA replication causes mistakes. So this is where you're going to check and see if you needed to make any repairs, or if you've accidentally duplicated chromosomes or made a bunch of errors. And if you do have any errors at this stage, this is where a lot of cells will basically go down. This is where we have apoptosis and everything, and like P53 is right here. So it makes sure that all the DNA is copied nicely, there's no mutations. So this whole process doesn't always have to happen so quickly all the time. A lot of times, and a lot of cells just spend time not dividing, and that's G0. They don't necessarily not have enough nutrients, they're not healthy or anything. It's just, they just don't need to divide. And this is, this is really good for a lot of our immune cells, like memory B cells, for example. Those are what make you immune after you do a vaccine. They essentially show up and they can revitalize your immune system when necessary, but they're not really like a huge thing that you need, for example, all the time. You don't want them active all the time so they can just like settle down like this. And you have plenty of other normal organ cells that you don't really need to divide over and over again. So this is just a closer look at S. So remember, think of this could be a maternal chromosome, for example, from the mom, and it's just one of your chromosomes. It happens to be maternal. It makes another copy of itself, and that's why sister chromatids means identical copy. This is maternal chromosome 7, this is maternal chromosome 7. We're not talking about paternal at all. And in that case, eventually we're going to separate at cytokinesis, or at anaphase, remember, we're going to separate, and we're going to split those 2 identical copies to 2 new cells. Perfect. So we kind of talked about this. We can't really study how mitosis happened. It might have been something kind of like an endospore where like the nucleus had to divide too, but we're not too sure. Endospores are just, they're neat because, these are really hard to kill in hospitals. It's not neat, but I mean, you should know it if you're going to work in healthcare. All right, we kind of talked about these checkpoints already. Really, you're just looking for size, nutrients, growth factors. Mechanical cues means like, okay, physiologically wise, is this cell a good cell? Like, is it shaped right? Is everything kind of working and where it should be, etc. like that. G2, this is your big one where the cell is definitely going to get a lot of scrutiny for how its DNA looks after you went through this S phase, remember. Because DNA replication isn't perfect, like we talked about. Maybe 1 every 20 nucleotides is mutated. You have a lot of energy mechanisms, or editing mechanisms, I should say. But it's not perfect, so sometimes G2, you need to make this cell go down if it's got a lot of damage. And then spindle checkpoint. This is just at metaphase. All you need here is just to know that everything's connected. See those kinetochores connected to the microtubules? The cell does not want to divide if one of these is not connected, because let's say this isn't connected right here, then that means that this double chromatid copy is going to go to 1 cell, and now you have 2 of the same chromosome, and that is not good for cells. Most genes are dosed to only be 1 or 2, so if you have this whole extra copy here, that's not a good deal. Okay. Go versus stop signals, this is just, you see all the different parts of the cell cycle. It's all this big fight between the go signals, the CDCs, CDKs, and cyclins that are going to be phosphorylating things, making sure that DNA is replicated, all that stuff. And the checkpoint genes all in red that are going to be saying, okay, is everything actually ready? Is there damage, for example? You can see that P53 is going to factor in nearly every checkpoint. That's why it's such an important gene. So essentially, these genes are going to fight, and eventually, if the cell divides, it means that the green ones accumulated enough cyclins, and everybody was happy enough to divide. So we talked about P53. There are lots of ways that it is going to be able to detect this damage, and that's really the way that it starts activating. And there is a way for it to start repairing DNA, but in reality, you're really going to end up killing this cell because it's much better to kill 1 cell out of 10 trillion than let a damaged cell go on. And the way it's going to do that is essentially just open holes in the mitochondria. This stuff comes out. Oops. All these caspases, remember the choppers, they get out of here. And then they have essentially chopped the cell to pieces. And that's cell death, essentially. So we kind of didn't go, I don't know if we went over this enough, but look at all this in, so let's focus over here to start. So say here's a cell and it's going through everything, and when you're kind of at synthesis and G2, you're probably, at this point, you're thinking of dividing. So cyclins, in purple here, those are those kinases. The more of those that you accumulate by turning more and more on, eventually you get to a point where you have enough of this cyclin activity that you will actually activate what's called MPF right here. And MPF is just kind of the mega gene, it's the no turning back gene. And as we can see here, if you activate enough cyclins, what's going to happen is you're going to shoot up with lots of MPF, and it's going to reach its massive amount. And that's all during mitosis, because remember, MPF is this key signal, mitosis promoting factor. It is the key to go forward in the actual mitosis. The main point of these cyclins and these kinases and stuff is to activate MPF. Once you activate MPF, it is pretty much a guarantee that you are going to go through the cell cycle. So you can see here the big deal is actually literally accumulating physical cyclins that are going to go on, combine with CDKs together, and they are going to phosphorylate and change a bunch of things. And eventually the big thing is that they attack, or they coordinate MPF to show up. MPF is what's going to happen and make mitosis really happen. This is just a good little graphic of how that's happening. Basically, cyclins and CDKs that start getting activated, they start phosphorylating all kinds of targets. These have a lot of different things. And remember that phosphorylations are usually going to essentially turn things on. So for example, in this little case, they're beginning the S phase. So it's really good here to just see like some of the things that they can do, and all of these things are things that we need to happen for that cell to split into two. So we actually didn't show this slide, but if you activate, so for example here in this experiment, if you activate, if you fuse cells together and one of them has all the signals that is making S happen, if you fuse them together, the other cell that wasn't actually doing anything, it becomes it starts replicating its DNA as well. So if you start sharing signals into one cell from the other, you're going to mimic that. Same thing happens here. If you have a cell, so on the right, this isn't dividing, it's not doing anything, but if you have one that's in the middle of mitosis, and you can see the little condensed chromosome here, the other cell is immediately going to share that MPF signal and just immediately start dividing. So that's how good MPF is that it can just literally physically transport over to another cell and cause it to start dividing. And we saw this with a little experiment. All you got to do is if you inject MPF, you start driving things right into M phase, and everything just happens all at once. We didn't actually do this either, but there are other growth factors besides MPF. Growth factors are usually just like really strong triggers that can cause cell division. So PDGF is a growth factor, kind of a hormone or a gene. And what happens is that if cells encounter this, so remember we saw signals can come and hit a receptor. That receptor causes a cascade of signaling, and that cascade of signaling has an effect. And in some cases, those cascades are the effect of proliferation itself. So if you have cells that are engaging and getting a ton of PDGF, they'll start dividing like crazy. So that's the end of one of the physiological pathways we kind of talked about, is that that can be your third cellular activity, is that the cell actually divides. That's why I think it was good to go over that lecture, is because that sequence of reception, transduction, and cell activity is going to apply to almost anything you see in physiology or cell biology. Okay, so we kind of went over this. When you imbalance your stop and go, you are going to cause a lot of issues. Namely, you're going to cause growth where there should not be growth. And sometimes it's only going to be one mutation, and this is only going to cause a small amount of growth. Now, the problem is that in this population, with one mistake, you can now easily make a second mistake. Now this population has 2 imbalances. It may get a third mutation, so on, so forth, until you end up with a population of truly bad cells that are out of control. In some cases, with a benign cell, they just activate a certain combination of stop and go signals that they're going to allow you to grow for a little while, but they ultimately aren't strong enough to make you or make a tumor that is going to evade any kind of therapy, or it's going to ever reach out to new tissues. There's just certain genetic pathways that have kind of a dead end. Another big reason why benign tumors show up and die, so think of like a mole on your skin, is like we talked about with that kill switch on the DNA, is that the ends of DNA, when you divide a ton, you're going to chew off the ends of DNA in these benign cells, and eventually you lose something important in all the cells and they die. It's malignant cells that have the potential to keep gaining advantages and keep gaining more and more and more mistakes. That's where you have an issue, and that's where you have to really intervene with aggressive therapy. Metastatic cells, these are the ones where they can go from the tissue that they originated from, like the lung cancer cells, and they can go find a new place, like the brain, like the, I don't know, like the stomach, I don't know where else lung cancers go. Like they basically settle into new organ systems, they start growing again, and they physically push out those organ systems and cause them to go haywire. This is the stage that kills patients. And this is the place that we want to avoid ever getting with patients, because at this point we don't have working therapies that we can rely on every time to get rid of them. So for the most part, most Go signals are fine in their normal doses. If you have a larger dose of a Go signal, though, it becomes an oncogene. Same thing, the stop signals are good to have. They're fine, they're normal, they balance out the Go's. But if you completely lose them, they are tumor suppressors, and you've lost a tumor suppressor if you lose them. So, I mean, you can always call stop signals tumor suppressors, you can always call Go signals oncogenes, but for the most part, there's a good balance between these two in normal physiology, so it's not going to matter. This is just a good graphic about p53. If it detects DNA damage, it's going to go and it's going to kill this cell. See apoptosis right there. So if it sees that damage. However, let's say this is one of your functional cancer mutations. If you've knocked out p53 accidentally and you have DNA damage, you don't have a working p53. Therefore, the damage is going to pass on to the next generation of cells, and it's going to keep happening over and over and over again because you have this unfixed damage that could have been halted by p53. So an example of a Go gene mutation is EGFR. There's a mutation in this region of the protein. Remember, so this is a receptor tyrosine kinase. See it? See how it comes together right here, and it gets phosphorylated here and here? Perfect, right? If you get a mutation here, these tyrosine kinases just come together all the time, and they never unbind. If they never unbind, they keep signaling over and over and over and over again down pathways like this and pathways like this. And the ultimate, so here's your transduction, 1, 2, 3, 4, and here's your activation. Transduction down this pathway, I'm actually very familiar with this pathway, 1, 2, 3, and then the final is cell survival. A lot of stuff in there. So that's why we covered a lot of those cell pathways, is that you can kind of see all the downstream effects that issues at the receptor can really have. We talked about this a little bit too, where here's your normal cell, you get one mutation. This population kind of grows around. Sometimes you get a population that gets one more tumor, but it's benign, it never really does anything. But eventually you get progressive mutations that eventually lead into some kind of really bad cancer, like this full one right here. And sometimes we can eliminate this, but the problem is that it can start from here again, go here, and you have a completely new metastasis that we have to handle a completely new way. Worst part is that sometimes things can come right back to this original founder cell and then go off and make some new terrible thing. Okay, so that's why a huge deal, and why a lot of our chemos they target mitosis, because cancer cells are undergoing mitosis a ton. So a bunch of chemos attack microtubules, a bunch of them attack cyclins, a bunch of them attack the centrioles, see that? and a bunch of them attack the kinetochores. So this is a good, so those of you that have watched this video this far deep, I'm going to tell you right now, I'm going to make a test question that is on something around these nature. If I make a tumor drug, what's one thing in mitosis I can hit, what's going to happen to it, and why is this effective against cancers instead of regular cells? Okay, so I think that's it. All right,